Hello everyone, I am Jay Berina. I am currently studying St. Mary's College, Autonomous Thuthukudi. In this video, I am explain about lambda-phage life cycle structure and application of bacteriophages. Life cycle of lambda-phage. This kind of bacteriophage are having both the type of life cycle, either lytic or lysogenic, both mode of life cycle. Lytic life cycle, what their actual goal is to bacteria replicated in the host cell and replicated and come up with all progenic virus. But in case lysogenic life cycle, they put their genome is somewhere that host or bacterial genome for certain period of time. After that, when they environmented in opposite to them, then the lysogenic will be converted into lytic. If they want stay longer time in the host cell, they choose lysogenic cycle. If they want a replicated in the host cell, they want lytic replication cycle. There is a very important molecule to switch all lysogenic cycle between their living. We will see and find it in the virus entered and delivered the genome, then DNA get replicated into protein and phage particle will come out by lytic cycle. But for the lysogenic cycle, they incorporate with the host genome and stay for several time. Now one important thing I want to tell you that the most of the phage particle or lambda-phage particle we are working, which is called as lambda papa. Structure of lambda-phage. Lambda-phage, a type of bacteriophage that infect E. coli, has a complex structure consisting of a head, a tail, and tail fibers. The head is an icosahedral protein shell containing the phage's double-stranded DNA. The tail is a flexible, non-contractable structure that facilitates host recognition and DNA delivery. The tail fibers or J chain are located at the end of the tail and play a crucial role in attachment to the host cell. Here's more detailed breakdown. Head capsid. The head is an icosahedral structure, meaning it has 20 triangular faces. It houses the phage's double-stranded DNA genome. The head is composed of capsomers, which are protein subunit arranged in a specific patterns. Tail. The tail is a long, flexible, and non-contractable structure. It is responsible for attaching to the host cell and injecting of the DNA. The tail is made up of protein subunit from the halo tube with a channel through which the DNA passes. Tail fibers J chain. These fibers are located at the end of these tail and are essential for host recognition and attachment. The phages interacted with the host cell, the tail fibers initiating the infection process. The specific protein GPJA on the tail interact with the lambda B maltodextrin porine protein on the E. coli cell surface. Genome. Lambda-phage contains a double-stranded DNA molecule approximately 48.5 kilobase in length. The DNA is packaged tightly within the head. The DNA is circularized upon entry in the host cell. Bacteriophages for food safety. The control of pathogenic bacteria causing foodborne diseases in an emergence of antibiotic resistance. Bacteria are important challenges for food industry and public health sector. In recent years, the single bacteriophage has biocontrol is being increased by accepted as natural green chemical free and antibiotic free and food safety to prevent current bacteria in food. What are bacteriophages? Bacteriophages that are phage or short of bacteria eaters that are virus. They're everywhere in the world and often consumed in our diet. They have food safety agent because phage are exclusively infect. The target bacteria it harmless to plant, animals and humans. They are extremely specific. Each phage can typically infect one of the strange or few strange of the bacteria within the same genus. They do not affect other bacteria present in the food, house or environment. Phages specifically recognize and touch it the bacteria, then they inject the genetic material, then they start producing the phage compound inside the bacteria rapidly. Only phage will reassemble and they produce an enzyme leading to a bacteriolysis and release of new phage and repeating the lytic life cycle again. As biocontrol agent, phage should affect in many times in target bacteria is possible a formulation of phage cocktail in complementary phages and target in different stage of origin antibiotic resistance is more even different species. Moreover, cocktail help to reduce emergence of phage resistant bacteria. Effective phage cocktail can be played different lifestyle of food chain to prevent growth non-desirable bacteria in the primary sector as a antibiotic to control infection in food production and in food processing environment. Bacteriophage control the food safety using the natural green and safety product.